Welcome back to Cancer as a Mitochondrial Metabolic Disease Basics. In the last video, we discussed how curcumin may help starve cancer cells by disrupting glucose metabolism. But there's a problem. Cancer cells have a backup fuel source, and that backup fuel is glutamine. When glucose becomes insufficient to support growth and survival, many cancer cells can easily increase their dependence on glutamine. In fact, some researchers believe glutamine may be just as important or even more important than glucose for driving cancer growth, survival, invasion, and metastases. So here's the big question. Can curcumin interfere with glutamine metabolism as well? The answer may surprise you. Research suggests that curcumin can suppress glutamine transport, glutamine utilization, glutamine-driven growth signaling, and even weaken two of the cancer's most important antioxidant defense systems, glutathione and Thioredoxin. In other words, curcumin may not only target both of cancer's major fuel sources, it may also make cancer cells significantly more vulnerable to oxidative stress at the same time. Today, we're going to dive into the science and explore whether curcumin can help starve cancer cells of glutamine. So without further ado, let's get into it. So as promised in the title and the intro, today we're going to be looking at how curcumin kills cancer from a perspective of glutamine and endogenous or internal antioxidant systems, or redox homeostasis. In the last video, we talked about how curcumin has been studied on all these different types of cancer, how it has effects on the PI3 AKT mTOR pathway, and we looked at how this PI3K AKT mTOR pathway affects glucose metabolism, and we then looked at all the places that curcumin can have direct effects on or effectively help shut down or collapse the Warburg metabolism or the fermentation of glucose into lactate. But in this video, we're going to look at how curcumin also affects the glutamine metabolism that cancer cells rely on. So let's reorient ourselves back to this classic C-free graphic here, where we looked at the left side of the graphic last time, how glucose is being converted into lactate, or the Warburg effect, or the substrate level phosphorylation of glucose into lactate to make energy and other things. In this video, we're going to look at the right side of the graphic, how glutamine is fermented to make energy and a lot of other things into succinic acid, and how curcumin could have a role at inhibiting that process. So the first paper I'm going to look at was published in July of 2024, and it was titled "Curcumin Synergistically Enhances the Efficacy Against Gemcitabine-Resistant Cholangiocarcinoma via the Targeting of LAT2 Glutamine Pathway." And it says here, in conclusion, curcumin synergistically enhances gemcitabine efficacy against gemcitabine-resistant cholangiocarcinoma by induction of apoptosis, partly by inhibiting the LAT2 glutamine pathway. And basically what we're seeing here is that curcumin is having a direct inhibitory effect, either by expression or direct inhibition. My gut tells me it's probably by expression, but having less of this LAD2 around means less glutamine can get transported inside the cancer cells, which then the cancer cells would feel as a metabolic stressor. In fact, curcumin just stop at this LAD2, also known as 7-8A8. It also has effects on the more classical glutamine transporter SLC1A5, has effects at the level of SLC7A11, the level of GLS or glutaminase, and GLUD or glutamine dehydrogenase. But those are all a bunch of big fancy terms. Let's look at them on an actual graphic to see what I'm actually talking about here. So unfortunately, this LAD2 is not shown on this graphic. It's a different glutamine transporter, but the more classical SLC1A5 right here is affected by curcumin. This is the glutamine transporter that I've talked about in the past the most, of compounds having the most effect on this particular transporter here. I also mentioned glutaminase, GLS, the enzyme that converts glutamine to glutamate. That's the enzyme that is directly inhibited by DAWN. Curcumin has an effect on GLUD, glutamine dehydrogenase. This is the enzyme that is directly inhibited by EGCG. And then lastly, we have curcumin having an effect on SLC7A11. This is in some papers known as the Achilles heel of the tumor. It is directly blocked by aristin or sulfasalazine, but curcumin has an effect here as well at probably downregulating this protein from being expressed or created. But that's not all. Curcumin also has effects at the blockade of an enzyme that is critical in the fatty acid synthesis pathway known as fatty acid synthase, FASN. And you see here that FASN although it does make new fatty acids for cancer, not for burning as fuel, but for raw materials, it also has direct regulatory effects on VEGF, or vascular endothelial growth factor, which is required to make new blood vessels, and other parts of the extracellular matrix metalloproteinases, etc. So curcumin has an effect on fatty acid synthase as well. But probably more in line with our main playlist right now, we're going over oxidative therapies in that playlist. It also has effects on antioxidant systems that cancer uses to protect itself from redox imbalance or free radical damage. And so one of the things that you may have missed in the prior graphic is this GSH here. This is a pretty busy graphic, so I can understand how you'd miss it. So GSH, I went ahead and put a little circle around so we can highlight this. Well, GSH stands for glutathione, and glutathione is made up of 3 amino acids: glutamate, cysteine, and glycine. And so where does glutamate generally come from? It comes from glutamine that's in high abundance, and then that glutamine gets converted to glutamate via the glutaminase enzyme. So glutamine directly contributes to redox balance, redox homeostasis, or antioxidant support for cancer cells. So you can imagine that if we block glutamine uptake or glutamine utilization, or we have effects on this SLC7811 transporter, we're going to have effects on the glutathione system, which is critical. But there's more. So curcumin also has effects at the level of the TRX, or the Thioredoxin system as well. You may recall this if you watched the main playlist on how cancer protects itself from reactive oxygen species, and the TRX system and the peroxyredoxin system was also highlighted in that. When we were talking about the TRX system, we focused on either TRX1 or TRX2 or just TRX as a whole. And we looked at a compound called aranophen. And I want to remind us of aranophen because I want to look at how it may compare to curcumin. So curcumin does not have the same exact effects as aranophen, but they work on the same system. So this paper was published in July of 2005 and it was titled Thioredoxin Reductase is Irreversibly Modified by Curcumin, a Novel Molecular Mechanism for its Anti-Cancer Activity. And it says here in the abstract, curcumin, which is consumed daily by millions of people, is a polyphenol derived from the plant Curcuma longa. This phytochemical has well-known anti-cancer and anti-angiogenic properties. In this study, we report that rat TRX1 activity and in TRX-dependent disulfide reduction was inhibited by curcumin. The IC50 value for the enzyme was 3.6 micromolar after incubation at room temperature for 2 hours. The inhibition occurred with enzyme only in the presence of NADPH and persisted after removal of curcumin. By mass spectrometry and blotting analysis, we proved that this irreversible inhibition by curcumin was caused by alkylation of both residues in the catalytic active site of the enzyme. However, the curcumin-modified enzyme showed a strongly induced NADPH oxidase activity to produce reactive oxygen species. That's what you call a double whammy. Inhibition of TRXR by curcumin added to cultured HeLa cells was also observed with an IC50 around 15 micromoles. Modification of TRXR by curcumin provides a possible mechanistic explanation for its cancer preventive activity, shifting the enzyme from an antioxidant to a pro-oxidant. So we see here that curcumin is having this irreversible effect on this TRXR. TRXR is inhibited, so therefore we have more ROS. And TRXR, if it's active, it can have downstream effects at boosting the antioxidant capacity of whatever cell it's in, including cancer cells. So let's take a look at the effects of curcumin, and these are graphics that I pulled from the paper, on the activity of this TRXR system, or this thioredoxin system. We see here it's a dose response. So at 0, its TRX is basically at 100% activity. And then as we increase the dose at 5 micromoles, we're about maybe 25 to 30% reduction in activity of this antioxidant enzyme system. And then if we get up to 10 micromole, we are now at darn near 75% reduced activity. And then it kind of levels out right around maybe, 25 or 30 micromoles. So the big question is, as with all things repurposed meds or supplements, nutraceuticals, is can we achieve these type of therapeutic, quote unquote, concentrations realistically? So let's look at this graphic comparing two different formulations of curcumin. We have a curcumin phytosome and we have a curcumin that's a liposomal formulation. And then depending on the dose, we see an estimated serum concentration of curcumin. And what we see here is that at 500 milligrams of curcumin phytosome in a single dose, you get a max serum concentration of about 0.27 micromole. What I would consider probably a max single dose for most people, 4,000 milligrams of curcumin phytosome gets us about 2.17 micromole. Now let's look at liposomal. 500 milligrams of a liposomal single dose is about 0.41. And if we go all the way up down to 4,000 milligrams for a single dose, we're looking at about 3.26 micromole. So where do we land on this particular graphic? Well, 5 micromole right here is getting us about maybe like 60 to 65% reduced activity of this Thioredoxin system. And if we go slightly south of there, perhaps 2.5 micromole, let's split the difference, 2.5, we're probably still inhibiting about 25% of the enzyme activity. But at the same time, we're unlikely ever going to achieve these like 10 plus micromole doses. And in fact, I had it calculate basically how many milligrams it would take to try to get 10 micromoles. And it would need about 18,000 milligrams of phytosome or about 12,000 milligrams of curcumin liposomal. Not only would that be extremely expensive, difficult to take that many capsules, you're probably approaching safe, tolerable limits with that. Now, the other potential option would be an intravenous dose, right? And so if you gave an IV infusion of curcumin, then we'd be looking at maybe like 1,000 milligrams of curcumin could approach this, and about 2,250 milligrams could approach the 20 micromole. But I will tell you that I personally do not think that in this case, these supra ability for oral curcumin to saturate Thioredoxin systems is ultra necessary. And why do I say that? Because curcumin doesn't just affect the Thioredoxin system. If you were only looking at it from one particular important mechanism, like it only worked on Thioredoxin, then it would probably be important that we only look at how can we achieve these higher micromolar quantities to try to have that activity. But we know that it has diverse effects. We've looked at it now at multiple levels, at signaling pathways, inflammation, glucose, and glutamine. And now we're just looking at a very zoomed in portion of a mechanism through this Thioredoxin system. And so I'm going to show you in future videos about why I believe that is probably not necessary to use 18,000 milligrams or 12,000 milligrams. It's probably not even safe at that range, to be very honest with you. But that being said, I don't think that you would need this much curcumin because there are studies that show that much lower doses have profound effects on mortality and survivability with cancer. Now, I did ask you earlier to remember this Aranophen compound that we have looked at previously when looking at Thioredoxin, the system. And we see here that the amount of Aranophen needed to, here's TRX activity again, right? So the amount needed to basically go to zero is somewhere between 0.5 and 1 micromole. Okay, that's a big difference, right? So to get nearly, 100% of activity, it needs 0.5 to 1 micromole, whereas curcumin to get, to be, let's say, as low as possible, this is about as low as where it starts to like kind of really flatten out here. You need 30 to 50 micromoles, right? Much more curcumin is needed to affect this enzyme system. And then I went ahead and did the calculations. So if Aranophen, if that was being used in a single dose, that would barely budge to 0.025 micrograms per ml. But if you used it for a shorter time, like a short burst, like 3 milligrams twice per day, then you're getting to 0.31 micrograms per ml, which ends up being about 1.5 micromoles, right? So then you're completely operational because now you're in this range for Aranophen. I just wanted to compare and contrast what a drug that basically only reason for being or main reason for being is this Thioredoxin system, what it's capable of at much lower quantities compared to curcumin, although curcumin in a lot of ways is much more impressive because it has many more effects. So in summary, how does curcumin kill cancer cells? Well, we looked at glucose. In this video, we looked at glutamine, and it has diverse effects on the glutamine system at the level of uptake, at the level of utilization, at the level of exchange to make glutathione. It is a glutamine blocking powerhouse. And that is why it's been studied so heavily and why it's such a powerful addition to many people's metabolic therapy protocols. If you like videos like this, please like, share, and subscribe. Until next time.